Hello students, welcome to this session. Let's study infection. Definition of infection: it is the process in which pathogenic organism enters, multiplies and invade normal anatomical barrier of host, resulting in disease. An infectious disease, when clinical manifestations appear, it is called infectious disease. Now let's see types of infections. So primary infection is the initial infection. Reinfection is subsequent infection by same organism. Secondary infection, subsequent infection by new organism. Focal infection, it is localized infection. It can be superficial or deep. Superinfection, the second infection in already existing infection. Cross infection, new infection acquired from other host. Nosocomial infection, it is hospital acquired infection. Heterogenic infection, it is physician or medical paramedical staff induced infection. Depending on source, the types of infections are endogenous infection and exogenous infection. So endogenous infection, it is from within body of host. Example, normal flora. So two types of normal flora: transient flora and resident flora. So transient flora is present at some point of time, not always, and resident flora, they are present constantly. It prevents permanent colonization of body by other microorganisms, that are pathogenic microorganisms. The exogenous infection from outside body of host, so that is environment, so air, water, food, hospital equipments like thermometers, stethoscope, blood pressure apparatus, bedpan, urinal, trolley, etc. Then healthcare workers, other patients and carriers. Types of infections according to the clinical manifestations. So they can be asymptomatic and symptomatic infection. Asymptomatic when no symptoms are seen, and symptomatic when symptoms are seen. So it can be acute infection when the symptoms are seen for a short period, and chronic infection when symptoms are seen for long period, that is more than 6 months of duration. Latent infection when infection is in hidden form. Types of infectious diseases depending on the epidemiological pattern. So it can be endemic, hyperendemic, epidemic and pandemic. So endemic infection when it is persistent in particular area at lower rate. Hyperendemic, the infection occurs at much higher rate than the usual in particular geographic area. Epidemic, when infection occurs at higher rate in particular area. Pandemic, when infection spreads to large areas of the world. Example, COVID-19. Healthcare associated infection, that is hospital acquired infection, is defined as new infection occurred in a healthcare facility by a patient after 48 hours of the admission, which was neither present nor incubating at the time of admission. Community acquired infection or associated infections refers to the infections which developed in the community or within 48 hours of the admission to healthcare facility. Some infections can be localized. They are present in local area. They can be superficial or deep, and they can be generalized, like bacteremia, presence of bacteria in blood; septicemia, presence and multiplication of bacteria in blood; pyemia, pyogenic bacteria are circulating in blood, characterized by development of abscess in the various organs. Source of infection can be endogenous, that is normal flora. It can be transient and resident flora. Exogenous can be environment or hospital equipments or healthcare workers or other patients and carriers. Sources can be animals, so they are called zoonotic diseases if the infection is from animals. So bacterial infection like plague, viral infection like rabies because of dog, then protozoal infection, toxoplasmosis because of cat, helminthic infection, hydatid cyst because of dog. So these are animal acquired infections. Then insects, they are called arthropod borne diseases. So they are vectors, can be mechanical vectors like flies, they cause dysentery, typhoid. Biological vector, they cause Aedes mosquito cause yellow fever. So mechanical vector when there are no multiplication or the fertilization. Then biological vector when some sexual cycle present in that vector is called biological vector. Then other source can be soil and water. So bacterial spores can be tetanus. Then parasites like microorganisms causing vibrio, hepatitis, viruses. Then food, it can be external contamination, food poisoning or staphylococcal poisoning. then pre-existing infection, meat, animal products. Carrier, a person who harbors infectious agent in absence of clinical symptoms but transmits infection via contact or secretions. So the types are healthy carrier without suffering from disease, convalescent carrier who recovered from disease and continue to harbor and shed pathogen from the body. Temporary carrier, they shed organisms for less than 6 months period. Chronic carrier, they shed organisms for longer periods. Contact carrier, acquire pathogen from patient. Paradoxical carrier, acquire pathogen from another carrier. Methods of transmission: contact, like sexually transmitted diseases like syphilis, gonorrhea. Indirect contact can be fomites, inhalation, droplet nuclei, ingestion, waterborne like cholera, foodborne, food poisoning, handborne, dysentery, inoculation, so tetanus, rabies, syringes, needles causes HIV, hepatitis B, hepatitis C because of the blood transfusion. Then congenital, it can be vertical transmission, syphilis, rubella, or iatrogenic can be because of the lumbar puncture, catheterization, transfusion, dialysis, or organ transplantation. Factors responsible for microbial pathogenicity: so they are adhesion, so because of the fimbria they are adhesion to the host cells. Invasiveness, so it causes spreading, it can be localized or generalized. Toxigenicity, so it is because of the endotoxin or exotoxin, this we will see in detail later slide. The plasmid can be R factor. Bacteriophage, so beta or tox plus gene, bacteriophage in diphtheria. Communicability, spread from one to another individual. Other bacterial factors like thrombin-like enzymes, seen staphylococci, fibrinolysins, hyaluronidase, leukoserines, hemolysins. Bacterial appendages like capsule or bacterial VI antigen in Salmonella, K antigen in Klebsiella. Biofilm is a group of bacterial cells which stick to each other on a surface. Example, prosthetic valve. Infecting dose is minimum inoculum size that is capable of initiating infection. Route of infection, any route or specific route required. So these are the factors causing microbial pathogenicity. Now we will see endotoxin and exotoxin differences. So endotoxin, lipopolysaccharide in nature, exotoxin, proteins. The source, endotoxin, form part of cell wall of the gram-negative bacilli, and exotoxin is secreted by gram-positive and gram-negative bacteria. The endotoxin released after cell lysis, exotoxin actively secreted by bacteria. Endotoxin heat stable, exotoxin heat labile. Endotoxin no enzymatic action, exotoxin action often enzymatic. Endotoxin non-specific, no specific tissue affinity, active very large doses and weakly antigenic, ineffective and not used for vaccine. Exotoxin specific action on particular tissue, specific tissue affinity, active in minute doses, highly antigenic, effective by neutralization antibodies, and toxoid forms are used as vaccine. Example, tetanus toxoid, diphtheria toxoid. Hello students, today we will have lecture on immunity. What are the points? Definition, types, it is innate or acquired, and then different types like adoptive, local and herd immunity. So first we will see definition. So it is defined as resistance offered by host against microorganisms or any foreign substances. Then what are the types? So it is divided into two types mainly, innate and acquired. So innate immunity or native immunity, it is the resistance by virtue of genetic and constitutional makeup. So it is present since birth. And then again it is divided into two types, the non-specific and specific. And acquired immunity, it is also called as adaptive immunity. So it is a resistance acquired during the lifetime. It is different from the inborn. And then it is again divided into two types, that is active and passive immunity. Then we will see difference between innate and acquired immunity. So innate immunity, it is present since birth, microbial exposure not required, it acts immediately, no memory present, and mechanism can be barriers like anatomical barrier, physiological barriers or secretory products in the body, and normal resident flora which protect from the pathogenic organisms. And it is divided into two types, specific and non-specific immunity. Then acquired immunity, it is acquired during course of life, microbial exposure is required, It does not act immediately. It takes some time to act to form the antibodies, and then memory is present for the second or subsequent infection. And mechanism is of by T cells or B cells, and it is divided into two types mainly, active and passive, and each is divided into natural and artificial. So now, what are the components of innate immunity? This we'll see in detail. So anatomical barriers, physiological barriers, then resident flora, phagocytes, natural killer cells, and complements present in blood. And the components of the acquired immunity, they are T cells, B cells, and complements. So now, this we already saw. Types of immunity, innate immunity is non-specific, and then it is divided into species, racial, and individual. And again, specific immunity, it is divided into species, racial, and individual. Now we will see factors responsible for the innate immunity. So it is age, extremes of ages. So extremes of ages, they have low immunity. Then hormonal influence. So endocrine disorders like diabetes mellitus, hypothyroidism, they increase susceptibility to infection. So that means low immunity. Then nutritional status. So malnutrition, this cell-mediated immunity, and hormonal, humoral immunity, that is antibody-mediated immunity, is low in malnutrition. The mechanism of immunity, innate immunity, so this is first line defense. So examples are, first is anatomical barrier. So skin, mucosa. So intact skin and mucosa prevent entry of microorganisms. The sebum present in skin, it is antimicrobial. The mucosa prevent entry of microorganisms. Mucus, cilia, they are present in that mucosa, propel microbes outside. Example, cilia in respiratory tract. Then coughing, sneezing, and urine flush, these all mechanisms, they remove bacteria. Then secretory products, they inhibit growth of microorganisms. So few examples are there. So in saliva, it is lysozyme. In tears, it is lysozyme. In gastric juice, it is HCl. Trypsin and protein digesting enzymes are present in intestinal juice. Then bile salts, they rupture the bacterial cell membrane and then thus cause the killing of the microorganisms. Then lactoferrin bind to the iron and inhibit bacteria. Spermine inhibit bacteria in semen. Sebum contains chemical toxin to kill the bacteria. Then physiological barrier, so temperature, which inhibit growth of the bacteria. Low pH, that is gastric acidity, inhibit growth of bacteria. So here you can see examples of non-specific defenses. So mucous membrane are physical barriers to microbes. Lysozymes present in tear and saliva. Then mucus coated hairs in the nose trap inhaled particles. Mucosa of the upper respiratory tract is ciliated, and that cilia in trachea help moving out the bacteria away from the lower respiratory passage. Flushing mechanisms like coughing, sneezing, lacrimation, salivation, peristalsis, urination, so they all help to flush out the microorganisms. Then gastric, so that hydrochloric acid neutralizes Helicobacter pylori neutralizes acidic pH, and that is why Helicobacter H. pylori grow in stomach. So blood complements iron binding proteins like lactoferrin, transferrin. So they all inhibit growth of microorganisms. Lysozyme, enzyme that degrades peptidoglycans. So gram positive bacteria are more susceptible than gram negative bacteria. Then phagocytes like macrophages are chief phagocytic cells. Free macrophages wander throughout a region in search of cellular debris. Then Kupffer cells present in liver and microglia present in brain are fixed macrophages. Neutrophils phagocytic when encountering infectious material. Eosinophils are weakly phagocytic against parasitic worms. Mast cells bind and ingest a wide range of bacteria. So these all types of phagocytes, they inhibit or kill microbes. Then now we will see acquired immunity. So acquired immunity is divided into naturally acquired and artificially acquired. And naturally acquired is again divided into active and passive. So we'll see example of active and passive. So active antigens enter the body naturally. Body produces antibodies and specialized lymphocytes. In passive, antibodies pass from mother to fetus via placenta or to infant in mother's milk. Artificially acquired active antigens are introduced in vaccines. Body produces antibodies and specialized lymphocytes. And in passive, preformed antibodies in immune system, in immune serum introduced into body by injection. So now we'll see comparison of active and passive immunity. So active is produced actively by host immune system. So host participation is required in active immunity induced by immunogen or infection. It is durable and effective protection is there, and immunity effective only after a lag period. So some period is required to build up the immunity, that is production of antibodies, so that is called lag period. In passive immunity it is received passively, so host participation is not required. Then it is in ready-made or form, it is transferred, it is transient and less effective. And immediate immunity, in that lag period is not there because it is a ready-made form. So in active immunity, immunological memory present, booster effect on subsequent dose is present. The negative phase may occur because of the previous antibodies and then not applicable in the immunodeficient persons. In passive immunity, there is no memory, subsequent dose less effective, but no negative phase and applicable in the immunodeficient people. Then mechanism of immunity. So in acquired or adaptive, there is third line of. Then we'll see types of acquired immunity. So that we already saw. Acquired immunity is resistance acquired during the lifetime. It is of two types, active, and then active is again of two types, natural and artificial. So example of natural immunity is following natural microbial infection, and artificial is because of the vaccines. And then passive, it is again divided into natural and artificial. So natural example is immunity transferred from mother to fetus, and artificial through the immunoglobulins or ready-made antiserans. Then naturally acquired, it is clinical or subclinical infections. We already saw response to a bacterial or viral infection. Example, chickenpox, smallpox. Artificial acquired active immunity, response to a vaccine of dead or attenuated pathogens. Immunity is provided by the vaccination. Then now we'll see a few vaccines. So vaccines are preparations of live or killed microorganisms or their products. So there are bacterial or viral vaccines. So and there are different types. So live vaccine, example of bacterial is BCG and viral OPV. Killed vaccine, example in bacterial is cholera, and in viral it is Salk, that is for the polio. Then subunit vaccine, it is typhi VI in bacterial, and viral hepatitis B. Then toxoid, it can be tetanus in bacterial. So we'll see a few differences between live and killed vaccines. So live vaccine advantages: single dose is sufficient, induces local and cell mediated immunity. It is more effective and long lasting, cheaper and used for mass immunization. Advantages of killed vaccine: stable and safe to administer, can be given in combination with polyvalent vaccine, no danger of spread of vaccine oriented infection. Then disadvantages of live vaccine: the reversion of virulence possible, can spread from vaccines to contacts, pre-existing viruses can interfere, cold chain has to be maintained, few cause local and remote complications. Disadvantages of killed vaccine: multiple injection, that is booster dose is required, and local immunity and cell mediated immunity is not induced. Then passive immunity. So passive immunity, when ready-made antibodies are transferred to a recipient, the recipient's immune system plays no part. It is called passive. It is only transfer of the antibodies or immunoglobulins. Then it is of two types, natural and artificial. So in naturally acquired from mother to fetus via placenta, or examples are breast milk, so it contains IgA and monocytes. Colostrum, it contains bifidus factors, antibodies that protect newborn from the gastrointestinal infections. Then IgG, a type of antibody that is transplacental and protects infant up to 3 months. Then artificially acquired from the injection of serum such as gamma globulin. The method of collecting an IgG from an infected immune person and transferring this immunity to an unprotected person, sometimes given to infants who are not producing enough antibodies. Example, ATS and ADS, that is anti-tetanus serum or anti-diphtheric serum. Next type of immunity is adoptive immunity. A special type of immunization is the injection of immunologically competent lymphocytes. This is known as adoptive immunity and does not have general application. Instead of whole lymphocyte, an extract of immunologically competent lymphocytes, it is called as transfer factor, can be used. This has been attempted in the treatment of certain types of diseases, example lepromatous leprosy. Then other types, local immunity, immune response at mucosal surface. Example, intestinal, respiratory or genitourinary mucosa, and it is of IgA antibody type. Herd immunity, it is overall immunity of community to pathogen. It prevents epidemic diseases. An example of herd immunity is following vaccination. So smallpox is eradicated because of the smallpox vaccination, because of the herd immunity. And then it can be example is DPT, MMR vaccines.
Hello students, let's talk about normal flora, normal microbial flora. So it is normally present in a humans either on skin or in mucous membranes. So it is called as normal microbial flora. So first definition, it is diverse group of microbial population that every human being harbors on skin and mucous membranes. So like skin and then mucous membranes like oral mucosa or vaginal mucosa. So there the normally few organisms are constantly present. They are called as normal microbial flora. So there are two types basically, resident and transient flora. So resident flora, these are the organisms are lifelong members of human microbial population. An example is E. coli, which have the beneficiary effects in a large intestine. Then transient flora, these organisms are for short period members of human microbial population and they may cause disease under certain conditions. So few beneficial effects of microbial flora. So they produce essential nutrients like vitamin K, vitamin B complex. So these are by the intestinal flora, that is E. coli. Then defense against microbial pathogens. So they compete with the pathogenic organisms and do not allow them to grow. So like vaginal flora contains lactobacillus, which provides protection against the gonococcal vulvovaginitis. Then prolonged antibiotic treatment alters GIT. So Candida species may cause diarrhea, Staphylococcus aureus may cause necrotizing enterocolitis, and C. difficile may cause pseudomembranous colitis. The bifidobacteria and base feeding bacteria, they inhibit colonization of enteric pathogens. So this is a beneficiary effect. The harmful effects of normal flora, when the normal flora are displaced from their normal site of the body. So if example, bloodstream infections caused by Staphylococcus epididymitis or C. difficile growing in gut after antibiotic treatment, that is clindamycin mainly, they cause pseudomembranous colitis. Or the in immunocompromised individuals like patient on immunosuppressive or diabetes or steroid treatment or cytotoxic drug treatment, this normal flora may overgrow and then cause disease, they become pathogenic. Now we will see factors which cause normal flora disturbed and then which promote the infection. So first is injudicious use of broad spectrum antimicrobial agents. So because of this use of broad spectrum antimicrobial agents, the intestinal flora gets normal bacterial flora gets vanished, and then because of that the pathogenic organisms grow. Then host factors like immune suppression or diabetes mellitus. So in immune suppression, because of the low immunity, maybe because of the patient on immunosuppression or patient taking cytotoxic drugs or AIDS, so all these condition, in that immune suppression also this normal flora gets pathogenic, which is transient in nature. Then physical destruction like irradiation, chemicals, burn, chronic irritation or minor trauma like in dental procedure, vigorous brushing. So this promote passage of Streptococcus viridans into bloodstream, and which causes bacterial endocarditis. So here you can see normal bacterial flora in some parts of the body. So throat is important. So in that, Streptococcus viridans is normal flora, Streptococcus pyogenes, pneumoniae, Neisseria group, then some Staphylococcus epididymitis. Then in skin, Staphylococcus epididymitis, Staphylococcus aureus. These are common normal flora. Then nose, Staphylococcus aureus, epidermidis, streptococci. In mouth, Streptococcus mitis. Then in lung, then urethra, Staphylococcus epidermidis. Then skin, like groin and perineum, it is same like skin. So these are few examples of the normal flora present at some locations of the human being.
Hope you find this session useful for your understanding and for your exams. Thank you.